are showing 3 different types of weighing balances, and depending on your requirement you should choose which balance you need, because every balance has its own range. Suppose this balance can maximum weight up to 820 grams and it goes up to 2 decimal point. So, if you can go precisely measure up to 2 decimal point of a particular sample. In this balance, the minimum range is written as 100 mg and the maximum range is written as 310 gram, and it can go up to 3 decimal point. So, like that, this one is a high precision weighing balance where it can go up to 4 decimal point. So, all this range will be written on that particular weighing balance. So, depending on your requirement, you should check first how much it can measure and how accurately or precisely it can weigh your particular sample. Now, suppose you are making some kind of buffer or media, then you can choose these 2 type of balances, because their high precision is not needed. Suppose you are measuring LB or trace, you can use this kind of weighing balance. But suppose you are experimentally designing a particular assay where you need to measure some inhibitors or small peptides or some kind of small molecules around a pH, around a value of 1 mg or 10 mg, then you should always go for a high precision weighing balance like this one, where you can precisely measure that particular amount. Those kind of measurement will not be happen here in these big weighing balances, where it will not give you the precise measurement. So, depending on your requirement, you should choose which weighing balance you need to use. Now, we will make 1 molar of 250 ml of trace. Now, what does mean the molar solution? So, 1 molar solution means 1 mole of particular solute is dissolved in a 1 liter of that particular solution. So, to make a molar solution, you need to know the molecular weight. Now, how will you know a particular sample's molecular weight? Either you can in online or net, it will be available, or the best way to do is to check the bottle of that particular solute or the sample. In that bottle, it will be written what is the molecular weight of that particular sample. Suppose here I am showing a trisma base, which is another name for trace, and the molecular weight is 121.14 gram per mole. That means 1 mole of trace weigh around 121.14 gram. So, if I want to make 1 molar of 1 liter solution, I will weigh 121.14 gram of this trace solute and then dissolve it into 1 liter of solution. So, it is a better habit to check the molecular weight in that particular box, because different sample might have a different weight, and always the online value may differ, because here I am showing EDTA. So, it is a common reagent for in biochemistry lab, and here the EDTA is dihydrate. So, the molecular weight comes around 416.20 gram per mole. But we have another type of EDTA. So, both are EDTA, but this is anhydrous, and the molecular weight differs and it becomes 292.24 gram per mole. So, suppose you are checking the molecular weight for EDTA and the online it is showing some kind of value, it might differ what kind of sample you have in your lab. So, it is a best practice to always check what kind of molecular weight it is written in that particular sample box and refer to that particular box, and based on that you should calculate how much you will need. So, I have mentioned that molecular weight comes in gram per mole. So, if you want to make 1 molar solution of trace in 1 liter, you should measure 121.14 gram and then dissolve that particular trace in 1 liter of solution. But we will make 1 molar of 250 ml of trace solution. So, for that there is a measurement. So, basically it goes half or 1/4 in this case. So, if you want to make 1 liter of 1 molar solution, you may measure that much of gram, 121.14 gram of trace, and then dissolve it in 1 liter. If you want to make 1 molar of 250 ml of solution, you just follow a unitary method and calculate how much trace you will need. So, here I have calculated how much trace I will need, and this is almost 1/4, and that will be 30.15 gram. Now, I will measure that particular volume or sample of this trace in a weighing balance, and then I will try to dissolve it in water. or basically deionized water. So, now I will measure 30.35 grams of tris to make 1 molar of 250 ml tris solution. For that, before we start measuring any kind of sample, first check the level of the particular weighing balance. So, all the weighing balance contains a small bubble which is a leveling bubble, and you should check if the bubble is at the middle or center of that ring, so that the weighing balance gives a accurate measurement. And also make sure that the weighing balance is properly cleaned. So, this kind of calibration or leveling is also required at a regular interval. So, after you are sure about the level of the particular weighing balance, then you open the sliding door of the weighing balance and you put the paper in which you will measure your salt. Now, the paper will have weight of itself, and then you need to tear the weight of that particular paper, so that the weighing balance make it as 0. Then when you measure your sample, it will show the sample weight, otherwise the additional weight can come from the paper. So, make sure you tear the paper or any kind of container which you are using for weighing. Now, I will gradually add the salt. So, as I have mentioned that I will do in 2 small batches to measure the 30.35 grams of tris. First, I have taken 15.35 grams of tris. I will take out this small volume and then put this sample into my clean beaker. Again, I will put this paper back into the weighing balance. Again, I will tear it to make it 0, and then again I will weigh another 15 grams of tris. Now, I have measured another 15 grams of tris. So, before you actually believe the value which is showing by the weighing balance, you should always close the doors, because if there is any kind of air current, it might change the readings. So, close the door and wait for some time and make sure the value is stabilized, and then you check the value. After you are sure about the value, then again you take out the sample, and again I am putting this thing in the same beaker where I have total 30.35 grams of tris. After you are done with the weighing balance, close the door. If there is any kind of spillage, you should immediately clean it with a tissue paper, and then you switch it off. Now that I have measured 30.35 grams of tris, then I will close the main container of the tris properly. It should be airtight, and do not use the spatula again to weigh something else. Always use a clean spatula, and you can wash it first with some kind of liquid detergent and clean it, and then use it for another round. The paper which we have used, if it is a reusable board, you can reuse it again. Otherwise, here I am using which is non-reusable. I will throw it so that people do not mistake it, and next time when you measure something else, you do not contaminate that particular sample with something else. After you switch it off, and then I have taken this 30.35 grams of tris. Now, I will pour some deionized water into it. For that, I have taken in a beaker some deionized water, and I have also taken a clean properly washed measuring cylinder which is of 250 ml of volume. So, as I want to make 250 ml of tris solution, it is not advisable to add 250 ml of water. So, you need to adjust the volume because the solution salt itself has some volume on itself, and again you need to adjust the pH of that particular solution. So, we will be adding concentrated HCl to adjust the pH, it will have again some volume. So, for that first we will add around 150 ml of deionized water. So, add some amount of water so that the salt is properly dissolved before you start adjusting the pH. Then after you adjust the pH, then make up the volume till 250 ml. So, right now I will be storing this deionized water, later we will be again needing this to adjust the volume. And here I have a clean dry magnetic stirrer. I will dip this stirrer into the solution so that it will help me to dissolve the salt. Now, I will put this container into a magnetic stirrer, and where it will make sure the salt is dissolved properly into the solution. As I have mentioned, I will keep the solution in a stir bar, magnetic stirrer. So, I have kept the stir bar inside the solution and I will switch it on, and it will rotate the magnetic stir bar and it will help me to dissolve the solute into the water. So, before we start measuring the pH, we have to make sure the whole solute or the salt is properly dissolved in the water. Now that the salt is dissolved completely in the solution, so make sure you do not put too much of water, also you should not put too less, otherwise the salt might not get dissolved. So, if you feel that the salt is not dissolving in whatever amount of water you have put, you can again put some amount of water more and then try to dissolve the salt completely before you go for pH change or check the pH. So, now that my salt has been dissolved in the water, I will check the pH of this solution, and around the trace pH is around 11 and I have to make the pH around 8.2. So, for that I will be adding concentrated HCl to change the pH from 11 to 8.2. So, for that, as I have calibrated my pH meter, I will take out the probe from the storing solution. And as I have mentioned earlier, always clean the probe with deionized water before you put the probe in another solution. Then dab it with a clean tissue paper to make the probe dry. Then open this cap before you start measuring the pH of a particular solution. Now, I will put the probe into the pH solution. Now, you can see I have dipped my probe inside my solution. Always maintain a certain distance between the probe and the glass wall of the beaker. Also, the magnetic stir bar should not hit the probe, otherwise it might damage the probe and you will get erroneous value of the pH. So, now the pH of this solution is shown in the monitor, it is showing around 11.48. So, to bring down the pH of the solution from 11.47 to 8.2, which is my desired pH of the solution, I will be adding concentrated HCl to the solution. Now, concentrated HCl gives fume, so you can do this in a fume hood, or otherwise if you are doing like me in a open space, maintain a certain distance from the concentrated HCl because it will harm you. And always use a glass dropper or pipette and add this concentrated HCl in very small quantity to bring down the pH of the particular solution. Now, do not put the concentrated HCl at one go, or do not put too much of concentrated HCl, or otherwise the pH will go down instantly. So, you should drop wise give the concentrated HCl and simultaneously check the pH of the solution. Now, the pH is showing stabilizing. That means it has to be stabilized before we can actually take that particular pH. So, as I dropwise adding the concentrated HCl, the pH of my solution is going down. So, we want to make the solution around 8.2 because the buffering range of the pH of the tris is around 8 to 9, and for physiological experiment or protein expression DNA, you need to have trace which is around pH 8.4 or 8.2. So, depending on your need, you need to change the pH of your solution. So, as I am adding dropwise the concentrated HCl, the pH in the pH monitor is changing, and as the stir bar is still there in my solution, it is constantly mixing the concentrated HCl into my solution, so that all the volume having a uniform pH. As you can see, the monitor is showing a stabilized pH value which is around 9.5. It is still not 8.2. I will add again small amount of this concentrated HCl into my solution, and then again wait for some time, and I will give the pH meter some time to stabilize the reading. So, until and unless the monitor is stabilized, the reading is shown as a ready value, we have to wait. Do not add too much of HCl at a one go, always add dropwise. Now that again the monitor is showing some ready values and I still did not get my desired pH, I will again small amount of the concentrated HCl and wait till the HCl which I have put into the solution is properly mixed into the whole solution and the monitor is also stabilized. Do not rush to make the pH at a one go, always add small volume and then wait. Be very careful while adding the concentrated HCl, it gives fumes, and always keep a water bottle or some kind of water beside you. So, if there is any kind of spillage or if you might get contact with the concentrated HCl, you just rinse it with clean water first. Now that I have got the desired pH of my solution, which is approximately 8.2. So, minor deviation from the desired pH around 0.01 to 0.04 is acceptable. So, I will stop here. Now, after I got my pH around 8.2, I will take out the probe from the solution, remove the solution from the magnetic stirrer, and again I have to wash the probe thoroughly with deionized water. Dab it with the tissue paper, and then for storage purpose I will keep the probe inside the storing solution again, and also I will make sure this cap is closed while storing, and then switch off the pH meter. Now, in this container we have 1 molar of tris which pH is around 8.2, but still it is not 250 ml. So, we have to make up the volume to 250 ml. For that I am again taking the same measuring cylinder and I am going to pour all my solution in that particular measuring cylinder without the stir bar. as you can see, while dissolving the salt, I have put 150 ml of water, but now the volume has been increased to more than 170. So, for this purpose you should not put the whole water at a one go. You should put sufficient water so that the salt is dissolved, and then after you adjust the pH, make up the volume with deionized water till 250 ml. So, make sure you use deionized water, do not just add any water you found, because then the pH also will change, and also it will put some other contaminants into your buffer which might be harmful for your sample. So, now I have 250 ml of 1 molar pH 8.2 Tris buffer. After this you can keep this buffer in a container, and which will be readily used by other purpose.
Hello everyone, this is Pritam Roy from Department of Chemistry, IIT Kharagpur. I am working under the supervision of Professor Swagata Dasgupta in Protein Chemistry Lab. I am one of your TAs, so hope we will come across various new and interesting experiments where we can learn various types of biochemical methods. See you soon. Thank you. We have seen how to prepare Tris buffer, that is Tris HCl buffer. For that we have taken powdered Tris solution in double distilled water, and to it we have added HCl dropwise and regulated the pH. Now, what was the exact problem out there? Now, in that case there was small confusion about the ionic strength. We have added HCl to it, but we cannot regulate the ionic strength of HCl out there. Why? Because we did not measure the amount of HCl we have added into Tris while doing the pH adjustment. Moreover, the HCl we purchase commercially, the exact concentration of HCl is not accurate all the time. As a result, the ionic strength of the individual components in that case cannot be accurately determined. Hence, the Tris HCl buffer preparation has some shortcomings. In this case, we will prepare phosphate buffer in a different method. For preparing phosphate buffer, we will take two different components of known strength. So, out here we will basically fix the strength, the concentration of the individual components. For that we will take sodium monobasic and sodium dibasic hydrogen phosphate. Now, let us see how to prepare the buffer. Now, the two components that we require for preparing this phosphate buffer are: number 1, sodium phosphate dibasic. This is sodium phosphate dibasic, one component, and the second component is sodium phosphate monobasic. Now, one thing we have to carefully note down while using these chemicals are its molecular weight. Now, the molecular weight are generally mentioned either at the bottom of this bottle or on the sideways. Now, here the molecular weight is written. The molecular weight of sodium phosphate monobasic having a molecular formula of NaH2PO4, the molecular weight is 119.98. Now, why do we require this molecular weight? Now, while calculating the concentration, that is the required concentration for preparing buffer, individual components have to be prepared of desired concentration. As we know that for making a desired concentration, we need its molecular formula. So, this molecular formula as well as the molecular weight is required out here. Now, this one is for sodium phosphate monobasic. Now, let us see the second component. The second component which we will be using is sodium phosphate dibasic heptahydrate. Now, in some bottles we can see this heptahydrate is not mentioned. However, we have to check out whether this heptahydrate is mentioned or not. Depending upon this, its molecular formula will vary as well as the molecular weight. So, if heptahydrate is not mentioned, if you see in any bottle where sodium phosphate dibasic is written, and the formula mentioned is Na2HPO4, then the molecular weight will be adjusted accordingly. However, out here, since heptahydrate, that is 7H2O is there, so the molecular weight will increase depending upon this formula. So, the formula out here is Na2HPO4 dot 7H2O. It is written here Na2HPO4 dot 7H2O, and the molecular weight written out here is 268.07. So, this is the molecular weight of sodium hydrogen phosphate dibasic. So, we have taken these two components, the monobasic one and the dibasic one, and now we have to weigh the amount of this components required. Now, before proceeding for weight, we have to calculate the amount of this individual component required. For that, we need to actually sort out the concentration of this component. So, the concentration of the buffer we require the concentration of phosphate buffer. The concentration of phosphate buffer we will be preparing is 0.1 molar or 100 millimolar. So, let us see the amount or the weight of this individual component required. Before proceeding for weighing this defined components, we need the spatula here, and the second thing we require is this butter paper. Now, before proceeding to spatula, I want to show you something about this butter paper. For weighing, what we need is this butter paper, and the butter paper should be folded accordingly. Here we can see we have folded it in a specific way. So, how to fold the butter paper? Here what we can do, we can basically fold it in this way. We can all the four sides we can fold it out here, and then diagonally we have to fold it. It takes something of this shape. So, basically it actually depends upon your convenience. Generally, if you do it in this way, the chances of this reagents or whatever solid substance we are taking of coming out or falling from this butter paper reduces when you take this sort of boat conformation or boat type of structure of this butter paper. Now, the second thing we require is the spatula. Okay, the spatula is a very common thing in a lab. However, it is very important for us to note down a very crucial thing, that is we have to use a clean spatula each and every time. Before using this spatula, we have to see if any impurities are there in the spatula or not. Visually, we can see actually whether any impurities are there or not. If any impurities are there, we can actually clean it using a tissue paper dipped in moist tissue paper dipped in water or double distilled water. However, the spatula I am using out here is completely clean and it has been washed with double distilled water. And however, before proceeding for the weighing, we are actually cleaning it using this Kim wipe. This part we are cleaning it. Now, let us proceed for this weighing part. So, we have taken this butter paper and kept it in this weighing balance. Now, we will press stir and here we can see it becomes 0 0 everything. Now, we will add the sodium phosphate monobasic. We have calculated it for 250 ml 100 millimolar sodium phosphate monobasic. We will require 3 gram of this chemical. Now, we will be gradually adding this one. Here we can see we have added just 1 spoon and it has become around 1.2 gram. Okay, so it is 2.5 gram. Now, once it reaches this 2.5 gram, we know that our desired weight is 3 gram. We have to add in small amount. You can actually better adopt a smaller spatula with a smaller mouth. We can get actually small spatula out here, But however, for large calculation, large weight like 3 gram or 6 gram, you can generally use this type of bigger spatula. Now here we are taking a very small amount of this sample out here and we are adding out here. Now we have to add again a bit. Again we are taking a pinch or a small amount of the sample. So better if we use smaller spatula, it will be much more convenient and easy to use. It is almost to 2.99. You can stop here, but I will take a pinch excess. Again a pinch of this sodium phosphate monobasic I have taken and I have added out here. Now you can question actually the weight I require is actually 3 gram and it has crossed from 3 to 3.032. So is there any error or not? Basically for small change in this weight, error won't be caused that much, but it is recommended to take some amount of excess weight. Why? Because once you pour this substance into a beaker or any conical flask or whatever thing you take, some amount of this chemical will stick in the butter paper. Generally do not stick in the butter paper, but if you use some other thing or any paper apart from butter paper, it generally sticks there. So the entire thing you have taken 100% does not come out. So better to use an excess of this chemical once you take any weight. So now we will transfer this one to a beaker. Now we will take this carefully. Now once you take it here, try to fold it in such a way, do not keep it flat, okay, so it will fall. So try to keep it in such a way and do not keep any beaker away from this weighing balance, keep it close to this weighing balance. So I am pouring here, I marked it basically this beaker and I am pouring here. Now we will measure the other part, that is, now we have measured monobasic, we will measure the dibasic one. Now we will measure sodium phosphate dibasic heptahydrate. For that we have taken this a clean butter paper, not the butter paper we have used previously, but a clean butter paper. We have teared it, the weight is actually 0.00 now. The spatula we will clean it, I have cleaned it actually previously after using the monobasic one. Now I will again clean it. Actually if the salt is, if any salt is attached out here, you can visually see it. However, I think it is free, no impurities are there out here. I am cleaning it once. Now the weight of this sodium phosphate dibasic required for preparing 100 millimolar 250 ml solution is around 6.7. So we will be taking 6.7 gram of this component. Here we are adding in parts using the spatula. Now we have reached 6.3, we have to add in small proportions. However, before proceeding, I want to discuss something out here, is that suppose the butter paper we are using for weighing purpose is overloaded with the chemical you are adding, you require actually. So what you can do is that you can actually note down the weight, transfer this part into a clean beaker and again weight and again weigh the remaining portion. Now if you actually take this overloaded butter paper, what we can do is that it can actually come out of this, it can fall from that butter paper. So basically in order to prevent that, you can basically take in 2 different steps, 2 or 3 steps. However, in this case we will be carrying out using this single step only, since I think it will not be overloaded out here. So, now we am adding in small proportion, as you can see out here, it is in small proportion I am adding, since it is 6.3 and the weight we require is a 6.7, adding in small proportion. Here we can see it has reached 6.726, however, an extra 0.026 or 25 gram we can actually neglect it, or it is better to take a bit excess. Again we will be taking out this butter paper carefully. Use both of your hands if you are not confident enough to hold it with one hand. Now take this beaker and add this one. So, here we can see we have taken 6.7 gram of sodium phosphate dibasic. This one is sodium phosphate dibasic heptahydrate, and once again I am reminding you, before calculating the weight, note down whether heptahydrate is mentioned or not. Now, what we have to do, we have taken this Na2HPO4 in one beaker and NaH2PO4 in another beaker. Now, we will add 250 ml of water and we will dissolve this using a magnetic stirrer.